So, good afternoon, everyone who are joining from Thailand and from other countries. A very good afternoon to all of you. Also, good morning to some of you who are joining from morning time zone, and also good evening to some of you who are probably joining from the evening time zone. It is actually a great pleasure for all of us here from AIT and AIT Extension to welcome you all to this very special webinar, which we actually consider as a free uh, learning sessions that is delivered and occasionally arranged by AIT Extension of Asian Institute of Technology. So this special webinar is on natural breeding techniques of mud crabs, which is Scylla serrata or Scylla species in household mangrove clay pond and its management in industrial scale. So just to tell you briefly about the webinar plan today, roughly this webinar will probably go ahead for 1 hour, which is 2 p.m. ICT or Thailand time until 3 p.m. But of course, depending on our discussion, question and answer, it might take a little longer. We'll see on that. My job here is to moderate the session. My name is Mohammad Zakir Hussain, or MD Zakir Hussain. I work for AIT Extension, which is the professional and executive training and professional services arm of Asian Institute of Technology. And I have together with me my executive director, respected Mr. Voravet Chonlasin. I will request him to say a few words immediately after my saying. And then, of course, I have a few other colleagues to help me. Thank you very much for all the colleagues, and special thanks to our speaker today for today's webinar, Professor Dr. Badrul Munir from UNISA, Brunei Darussalam. So just to bring you all to this webinar, I believe and I can see from the participant list that there is a very significant number of people who have registered for this webinar, and we understand they are very much interested about the topic in a specific to crab farming and crab breeding probably. So we will try to bring a little bit about in the context that why we are doing it and what is the reason behind. As I said, we are representing AIT Extension. Please, those of you who are actually representing agricultural domain or with agricultural background, do not please think that we do agricultural extension. We call ourselves AIT Extension, which is basically mandated for extending the AIT's academic and research services to the wider audiences. And that includes professionals, farmers, students, researchers, scientists, and also business people who are probably interested about the topic of our webinar. Our main mandate is to do the non-degree training and professional education programs, but we also are responsible to conduct consultancy and the research in very specific areas. And some of us are doing it in many different areas. Probably my executive director also will introduce AIT Extension briefly. Now, I'm representing AIT Extension as like my executive director and my colleagues, Mr. Kona Kiron. But today's speaker is actually from Brunei Darussalam representing UNISA, which is actually University Islam Sultan Sharif Ali. In short, I think we call it UNISA from Brunei Darussalam. I will introduce him with little more details after our executive director say a few words about the webinar. Now, as I said, I like to say a few things about today's webinar, which is about mud crab. As you probably understand that there are a lot of many varieties of crabs globally. I think we understand there are more than 4,500 different species of crabs globally distributed. But among them, very few are having very high economic importance. And Scylla species or Scylla serrata and other 3 species are the most important crab species which are playing a great role for the economic development, poverty alleviation, and the livelihood generation of many of our coastal communities and farmers. You know, why we're doing it? AIT Extension is actually occasionally arranging training and capacity building programs, and that could be in the field of aquaculture technology, aquaculture management, or even integrated coastal zone management or coastal ecosystem management. Within those training programs, we are actually bringing these modules where we basically try to bring the knowledge and expertise that we need for the crab farming for the coastal communities. People who are living in Southeast Asia, including Thailand, of course, they are very much familiar with swimming crabs that we all know. But you know that swimming crab is something that we cannot farm. But the mud crabs is something that we can actually farm. And there are historical evidence and data that suggests that there are countries like Southeast Asia and South Asia, people in the coastal area, they are farming the crabs in the pens, in the ponds, and in many different kind of structure that they use in the coastal area. The main way of doing this farming was basically they are collecting the fingerlings, the baby crabs from the nature, and then bring those baby crabs in the ponds, and then of course rearing those or fattening those, and then basically selling it. You can probably also understand, those of you like to eat crabs, that how expensive this is in the market. You know, economically, crabs are considered to be the third largest crustaceans from the sea that people consume, after shrimp and after lobster. So after shrimp and after lobster, crab is the third largest economically important crops that actually we can get it from the sea and from the coastal areas. And it has been actually widely practiced, not only for the catch, but also for the farming. But one of the challenges for the farming or aquaculture for the mud crab is the baby crabs. You know, traditionally we have been catching these baby crabs from the coastal areas, which is not very sustainable practice, and it is also not very abundant. And there are many reasons from human intervention, climate change, and many other reasons. The population is very decreasing very much day by day. So it is very much required that we go for breeding of the crabs in order to fulfill the farming requirements. That is why this session or this webinar is very, very important. So today we are very fortunate to have our speaker who has done wonderful work in this area. He have had projects in Bangladesh, and he has been teaching in Malaysia and Brunei Darussalam using his practical experiences of doing natural breeding of mud crabs. So he will basically explain his experience and the projects that he has been actually leading for years probably, and also the students' research that he has also guided. I think with that, this is the time that we like to proceed with the next session, which is actually welcoming our executive director. We are very fortunate to have our executive director, Mr. Boravet Chanlosin, to have with us. Thank you very much, sir, for your time. We wish you to say a few words and open this webinar for all of us.
Thank you. Thank you, Dr. Sagir. Good afternoon, Dr. Mooney, and my dear participant to this today's webinar today. Is very, very interesting one. It's very technical one. And as you know, that AIT Extensions working with the experts from around the world to transfer the knowledge to all the audience and participants who are interested in the subject area that they are keen to do businesses and also to do the knowledge transfer. So our program is not limited to the academics and also the industries, but we are also focusing on the new businesses and also entrepreneurships and small businesses that can give the opportunities to our global citizens, our clients, the organization in your countries to be able to start the new business or to enhance the business that you are doing, and also the farming business and also agri-businesses. So the subject today, although it is very specialized and highly technical, but nobody can say no to the opportunities to leverage from this, the mud crabs that we can really make the advanced research and advanced study, and also make it into the the industrial scale, and also into the business in the futures. And in many countries that have the resource available, can also initiate this business into their areas, in order to raise the income for the local communities, and at the same time it can also expand into the global scale, where this industry can go beyond the domestic industry, but can be also the export items to other countries. So with that, I would like to open this webinar with the expert from Brunei Darussalam, and I thank him very much for spending his time with our audience today. So without further ado, I would like to welcome you all to this webinar on natural breeding techniques for mud crabs in household mangrove clay ponds and its management in an industrial scale. Thank you.
Thank you very much, Achan Borabe. Thank you very, very much. So before we proceed, before I introduce the speaker a little bit, let me also request all of our audiences, all of our participants to feel free to ask any questions. I think you might have the option called Q&A. So you can actually keep writing your questions that you have been hearing from our speaker, and you have been, taking the notes, and then based on your notes, you might have some doubts you want to ask. So keep writing your question in the Q&A box. And also, if the chat box is open for you, we can also write there. So basically, our plan is to introduce the speaker first, and then invite the speaker to start his presentation. And after the presentation is over, we will go into the Q&A session, where we'll try to address all of your questions. And at that time, if you have also the option to raise your hands and other things, you can also instantly ask. But one request will be, when the presenter is going to present, maybe we will not entertain any instant question at that time. So with that, let me introduce briefly to our speaker today. As you have heard, Professor Dr. Mohammad Badrul Munir, he is now working in the aquaculture program under the Faculty of Agriculture in University Islam Sultan Sharif Ali, which is in Brunei Darussalam. His academic career actually started from University Malaya Sarawak, or UNIMAS of Malaysia, where he actually worked since January 2017 until July 2023. And then, of course, from August 2023, he actually moved to Brunei Darussalam, and he has been working there. Dr. Munir is having a PhD in aquatic animal nutrition, specifically on dietary probiotics, prebiotics and probiotics from University Science Malaysia, which he actually has completed in 2016. And he actually, before that, finished his Master of Science from Asian Institute of Technology. So we are very happy. Actually, he is our alumni. He did his master's from Aquaculture and Aquatic Resource Management program from AIT in 2003 actually. Before that, of course, he had his previous education from Bangladesh. He also had another master's and a bachelor degree on fisheries and marine resource technology from Khulna University in Bangladesh. And I think after his bachelor and master's, he was also working in Bangladesh before moved out of the country. He was working with the Danish funded or supported program called Danida Technical Assistant Program in Bangladesh for a couple of years. He was also working with the fish and seafood industry when I was working there as well. So we found each other as colleagues, and also like a good friend. And also it is good to mention that we have actually studied together in our bachelor degree. He also has spent a couple of years in Denmark. He was a Danish citizen for some time, but unfortunately, probably he didn't like the weather. So he moved back to Asia from Denmark, and then he started his second life or third life in Malaysia, and now in Brunei Darussalam. I think with that, let me talk a little bit about his research interest. Dr. Munir is having his keen interest in sustainable aquaculture, which includes aquaculture, its system and management practices. He has been teaching and doing research on that. He is also an expert on fish nutrition, immunobiology, and post-harvest technology. He has actually published 2 books, 14 book chapters, and 53 scientific articles throughout his academic career. He has 3 intellectual property rights, which is very interesting. We may like to hear about that at some point. And his first IP or intellectual property rights was on biogenic fish feed, which is very interesting, commercialized by a Malaysian private company. That's wonderful to know, Dr. Munir. Apart from his research work, he has also developed 2 MOOC courses for the global aquaculture communities. I believe you know about these are open source academic resources, which actually people can use it. So he has developed 2 such courses in the MOOC environment or MOOC ecosystem. And he has, of course, presented his research findings in many, many conferences. So with that, it's a great pleasure and an honor to welcome Professor Dr. Badrul Muni to start your session, sir.
Good afternoon, and for all of you, my salam. Especially Dr. Zakir already explained about me, myself, what I do. Actually, I am very much proud that I am also a graduate from Asian Institute of Technology, Thailand in 2003. Then I do many works, and then at last I do my PhD from Malaysia. Yes, today this is this was a very much sensitive project. When I get this project from United Nations Development Program, this project I achieved when I work in University Malaysia, Sarawak. It's a big competition. Then after that, I got this project because natural breeding actually sometimes people not understand how social natural breeding, because natural breeding is not like induced breeding. You not force the mud crab or any males, aquatic animal, not forced to do breed. This is naturally they do. This technique, how we do, that's today I am going to present. And it's a very much interesting. Honestly, it's very much interesting. So I am sharing my lecture slide. Everybody see my presentation?
Yes, sir, we can see your presentation. Thank you.
Okay. So this is the project name is Natural Breeding Technique of Mud Crab, Scylla sp in household mangrove clay pond, and its management in industrial scale. You can see here I not give the name of the species, Scylla sp, because in Bangladesh, where it's breed, they also not sure which species they usually do for breed. So that during this project, we also confirm what types of species it is. Because Bangladesh is 2 species usually available, Scylla serrata. Scylla serrata, one of the, but it's in the other in the Chittagong zone. But our one is different species, so I can give it this one when I go the presentation. And before going this, because it's a household mangrove clay pond, so this is one of the interesting terminology. That means clay pond. What do you mean by clay pond? Usually in Asia Pacific region, actually in Bangladesh there are lots of pond in household, and usually if I use this terminology pond, they can easily understand of this pond. Where I perform this work, every farmers have 2-3 ponds, big ponds there. So that's thing. What is it? To answer this question, I have 2 questions. First off, is it general household pond? What do you think about this? This is general household pond. That means is a regular use for household purpose, that pond? The answer is no. It's a pond or a series of ponds situated near the household. That is we call household pond. Then another question comes, because this is mangrove. Is it pond situated in the mangrove forest? This is one of the environmental threat, because if you excavate pond near the inside the mangrove, you must cut some mangrove forest. That's also environmental threat. So the question is, if it is mangrove forest pond, so what is that? What is this pond? Actually, this is not situated in the mangrove forest. It is a pond or a series of pond situated near to the mangrove environment. Mangrove environment, when you see the soil, this soil is different than other this is different than in soil quality. You can see when you see the mangrove soil and the natural soil, normal soil, this is some different. Mangrove soil usually black, and then more of organic matter there. So these are the mangrove environment. And you planted mangrove trees like in a Bangladeshi, this name is kakra, local name, and vine. I also give it the picture of which one usually known in Thailand, Malaysia, and this belt. I think this is not too much big mangrove trees, is a small like a garden trees, this one. And it should be planted in a zigzag pattern. Zigzag pattern means just crossing zigzag pattern. And then why is need for the betterment of the pond water to direct connection to the sunlight and wind. That is the reason zigzag pattern we needed. And this is the mangrove household mangrove clay pond. I think I give the picture. You have already understood what is mangrove or the household mangrove clay pond. And our project also we have established like this type of pond. And this is our actually project pond, this one. And this is we call vine. Bangladesh is called vine. But in Thailand, I think it's called upati. Upati, local name. And the scientific name is Abyssinia officinalis. So I have also given here some local name for your better understanding. If you want to do this thing, it's a very easy technique. And if you, when you planted it, you can try to find out these trees. And another one is kakra. Is Bangladesh name is kakra, but in Malaysian name is bakau besar, bitat, timbas, tumas, and Bangladesh name is kakra. Scientific name is Brugnia gymnohija. This one. So this is not a big mangrove tree. Usually, if it's a mangrove, then you are thinking this is big tree. This is like a garden like. And you see here, if it is long term, this is our project area, and we also planted this thing. Then the second is mud crab. Already you know there are many more than 4,000 crabs in this world. So which crab is the one? The mud crab is commonly recognized mangrove crabs under Opportunity family and Scylla genus. These are the important. We usually known as this is the mud crab. And basically there are four mud crab. These have an economical and potentially important. Usually scientists already success to breed. And these four is Scylla serrata, Scylla olivicea, Scylla paramomsian, and Scylla trancubarica. In Thailand, this is very much interesting that Thailand is blessed to have all these four species of mud crab. So that when I was in AIT, also I also visited some mud crab farm. That time I don't know actually how to culture. I still learn that time. So I also see there are many conventional hatchery there. This is concrete based hatchery. And they mainly breeding Scylla serrata. In this project, I already mentioned in the title Scylla sp. That is species name I not mention. This species in Bangladesh, we identify this is Scylla olivicea. And the most interesting fact is, when I started the project, especially the local farmers and the BFRI, Bangladesh Fishery Research Institute, they also informed us it's quite difficult breed using local mud crab. And that's time actually when I got the project, I also little bit nervous. Actually, how possible if local mud crab not breed? But I don't actually not not taking too much note about their what about their experience. I just asked my two students. They are the PhD student. I just say you collect the mud crab, and then we see really this is breed or not. If not breed, this is also our research. We can find something. If not breed, this is also our result. So we can try. And our one of the private organization where we adopt this technology, he also assured me if local mud crab not breed, he will bring it from the Chittagong, that is Scylla serrata or Scylla olivicea. But I didn't agree. I just say first I try, I catch the brood from the local river and I see what happened. Because my trustness more because which feed, I think you already know I have one IP product that is biogenic fish feed, that is probiotic feed. And this feed, when I apply the difficult fish, this also breed, especially ikan haruan. Ikan haruan is the name of Malaysian name, but in Asian snakehead, that one. Also, I apply it crustacean, especially udangala, that means prawn, Macrodicum rosenbergi, and then also Peneus monodon. I also apply. That's a very nice result I got. So that I just advised to my student, please collect the crustacean local mud crab, rear it in pond, and then you feed biogenic feed, and hopefully it will be breed. But that is just my assumption. It's not any result. But that time I faced some difficulties. I just share it. Difficulties of this. Some of the technical person, they are already consultant of that area in mud crab. They just say mud crab never eat formulated feed. But I keep my decision that I must use the biogenic fish feed, and that I try to feed the crab. That's actually my challenge. And Alhamdulillah, I also success for that one. So that one I can show you this presentation slide and you can understand this one. So I already told you, in Thailand, there are a lot of conventional mud crab hatchery. I don't know the statistics of that one, how much, but most of the Thailand is the good for mud crab production so far. But the mud crab conventional hatchery, what their problem? Their main problem is because mud crab is a highly cannibalistic species. That's one which have global attention, usually survival rate from Jua 1 to a small crab range 0.6 to 7.9 with a mean 3.3%. But it's a very much control condition. You can get it 3%, but most of the cases who are practicing in the commercial purpose is very difficult to get more than 1% survival rate. That's one industry actually not too much profitable. That's one, and sometimes they not get a single crablet also. So that's the reason, because this is a highly cannibalistic, and the conventional mud crab hatchery, they usually use hormone for induced breeding, concrete tanks for crablet rearing, use formalin to disinfection, use antibiotic oxatetracycline, azithromycin to free from pathogen. These are usually practiced in the conventional hatchery. So what the impact of the antibiotic? I think this is my supervised PhD student, Abdul Hanan. He also performed one comparison study and he will present that one. But I just say a little bit, because when human, if you are a human, if you take when you are sick, especially cough time, then you use the high antibiotic. If high antibiotic when you use, your total, the impact of the antibiotic also react your body, and then you not perform anything in a normal way. Also, you are feeling a stress if you use the high antibiotic. Same thing, when you cannot say the mud crab is like a aquatic species. It's no have any live value. It's also live organism. First, you consider this is also live. Although this is not a human, but it's alive. It's alive, and then it's a live crustacean. So when you give the inject the antibiotic, its body creates some stress. So this antibiotic and these chemicals you give it in the brood. That time the mother or brood makes more more stress. So if the female body, they feel stress when they produce baby, that baby also transmits some stress. And when stress, their cannibalistic nature is more. And then when it's grow from Joa to a small mud crab, different stages, because each have a Joa 1, Joa 2, Joa 3, Joa 4, Joa 5. Then different stages when it's pass, the cannibalistic nature implemented, and that time the number of crabs, they are eating each other, and the number of mud crab, they are going to less and less and less. Once upon a time is less than survivability. If it is commercial scale, less than 2%, but most of the cases is a 1% or sometimes zero. Some I also found in Malaysia when I work in Unimas. That time I also visited one of the mud crab hatchery. They also said they are fighting for survivability. Sometimes their survivability is less than 1%. Sometimes they not get a single crablet. Because of this, I just assume that this is of this, because stress mother should not be produce healthy child. And stress makes the mud crab automatically, this is highly cannibalistic, makes more cannibalistic. That's the reason. Although they free from the infection, but they are attacking each other and then eat. Also, you are supposed to know the mother brood also, when it's bigger, it eats its own babies also. So that's the reason conventional hatchery is not that is successful. Nowadays, aquaculture scientists or aquaculture scientists who are really interested in mud crab. Yesterday, I also reviewed one of the paper in the aquaculture journal. There they do hybrid mud crab production. That means they want to reduce some of these their cannibalistic nature, so that they do hybrid. Hybrid means which one is the less cannibalistic, which one is the high cannibalistic. They're just crossing. This is hybrid they're doing. I also review that one. So nowadays scientists more attention of mud crab, because mud crab has a potential economic important, that one, especially in Asia Pacific region. So my question is, if it is really Thailand have all 4 mud crabs, and as well as it's a very easy technique, or you can understand after ending my lecture side, is it really easy or not? That time, why not Thailand adopt natural breeding in household mangrove clay pond? That one. So anybody knows how to breed mud crab in nature? When I think you can asking when this is the natural breeding. So my question is, have you seen when mud crab breed in the natural water body? How deep they go? So this is automatically natural breeding. That means by Almighty Allah perform this breeding actually, without human interaction. The mud crab is breeding in the natural water. So that water, if they breeding, if they are survive, why not we do it in the inland? That is the thing. So this is the natural breeding. Natural breeding means without interaction of the human. Natural without human interaction, and this is natural breeding. So when I go for implemented the natural breeding, my student and my team also survey to the natural breeding ground of the mud crab. What about their water quality? What about their total environment? And when they find out the feature of natural breeding ground, we try to make same environment to the inland. Same environment. That means also they like the dark environment. They like pinpoint silent when they're breeding. This type of environment we will create. And when we create this mangrove hatchery, we also declare in a sound, nobody allowed to enter this natural mud crab hatchery, because it should be pinpoint. And then when brood may become mature, it needs silent environment. And we also tested different salinity, because when they are survey in the breeding ground, they get different salinity, 24 ppt salinity, 26 ppt salinity, and some 28 ppt salinity. So we also tested before starting breeding. We also tested which salinity is optimum for mud crab breeding performance. We got 26 ppt, and that one we maintain all our pond 26 ppt. So natural breeding is without human interaction. You can define natural breeding in two ways if you establish it is in your household pond. Number one, natural breeding of spawning takes place in a pond with little or no outside intervention. That means you just you just release the brood to the farmers. It when it's calm, natural time is naturally breeding. No need to you force them. Just like our human. I just give the example of the human. When human, our woman give the child, that time if it is natural, just have a counting some time. Then when time comes, automatically new baby is coming. These are the philosophy of this natural breeding. You just do. This is a pond, and then you just no intervention. You just manage. You just manage the brood, because when you brood, when you first egg come, it's like a orange color. Then when it's mature, it's turned into orange to black. Then when it's blackish, you can keep it in the pond. Automatically, it's release the when it's hatching. All eggs I also give here hatching, and you must keep your patience. Don't be too much. We do very fast work. You just give the time to the brood how it's hatch in the pond, naturally hatching. That is called natural breeding. And natural breeding you can also giving in aquaculture definition is an induced spawning without hormonal treatment by simulating natural spawning condition in the ponds or tank. That means you not give any hormone, you not give any antibiotic, you not give any chemicals, not force to the brood mud crab to breed. That is meaning of the natural breeding. You just see this is our project mud crab. You just see this is the egg already, and when we see when you see it turns into black, and when it's come for hatching that time you can easily understand the crab is sometime moving. They want to move some interesting characteristic they show, and that time you can keep it in the box, or because this is our project, so we need to submit to the UNDP how about the survival rate, and they also give us some specific result they want to see, so that we keep this brood to the pot, and then we see what about, but we not give any chemical, just to wait when they are doing hatching, and that is the hatching. You can see this is hatching, already hatching. This one in the we just keep it this, and then after it takes at least 2 hours. And my project, my Abdul Hanan and the project staff, my another PhD student Jahidul Hassan, they also wait, and they're not talking. You not see here, there is no sound. It automatically you see the already mud crab already given the eggs release already hatching release. You can see it's a million of this number. So just you wait until there's just like a natural breeding, not forcing anything. This thing, I think everybody you see, like, right? This one?
Yes, it is
This is the
working.
hatching.
Yeah, it is
This is
working.
the hatching. You just see he is now in Italy. Who is the field staff? He is now in Italy. He actually, I really acknowledge to him in because hatching have no any fixed time. Either it is morning, or it is lunch time, or it is midnight also. So he all time 24 hours, 7 days during this time and seasonal breeding time, he stay in the near the pot. Then our UNDP have given us target for 4 cycle. So that 4 matured brood, we just collect and we just keep it in the pond and just waiting how long it go for hatching. So that one we just do. That result also I produced today. And then this is how to perform. Perform already I mentioned. Establishment of a sustainable model of natural mangrove-based mud crab hatchery in household clay pond. My information is that if we really want to do mud crab, no need to a specific conventional hatchery. If you have a pond, which contains salinity 26 ppt, temperature is optimum 26 to 28 degrees centigrade. That one, when you maintain this, you just collect the brood, mating grab it. You just take it from the nature, then keep it in a box, bamboo box, because Scylla olivicea, Scylla serrata is a active cannibalistic. So that to protect from cannibalism, you just make one box, one crab, one grab it, then you feed them. Then when it's calm for hatching, it's automatically is produced. I have at the last site, we already made a animation video, but it's quite difficult to make the natural video because we don't know when it's go for hatching. So we not do any eye ablation. It's automatically is breed. And then what the benefit of this? When is breed in the pond, you just remove the box. Remove the box with the or the grab it mud crab. Just place it in the broodstock pond. Broodstock pond that place, and then you just give the time. A certain time, you can see after 26 to 28 days, you will see some crablet. This is one of our experience. When we first breed, we release all the 50% we just take it in our laboratory, and 50% we just release it in a pond. And number 1 day, number 2 day, number 10 day, number 12 days, we didn't see a single mud crab. And my PhD student and my affiliate, they also audit. Maybe we not success because we only release for 50%, 40 to 50% we release in the pond. That one. Then after 28 days, they inform me when it's come from the 26. I think this is more authentic information given by my PhD student. 26 days, when they go to the pond, they say lots of small mud crab. And so that one we show this presentation slide, and that's time we confident that yes, local mud crab breed. And we use local mud crab. I always repeat, we use local mud crab because our project is the southern part, is near mangrove. I think all of you know about Sundarban. This is the largest mangrove forest area. So that area, we just collected the local liver, that mud crab. We never adapt any imported mud crab. These are from the local we collected. So these are the project basic. Just we signed the project in UNDP November 2021. And this is first time we contact only 1 year. 1 year project. Then 1 year project actually when we constructed pond, when we do survey, and then we lost the breeding season. Breeding season because when you do the natural breeding, I forget to tell you, you must respect the breeding season of the mud crab. Then when we start the project November 2021 and October 2022, this time we lost because of the construction of the pond. Then some cyclone effect, because Bangladesh, you know, is a problem of the cyclone. And that time we faced 2 big cyclones. That's why we didn't adapt my full method. All this, so that we just breeding just naturally. We just collected some gravid mud crab, and then we just breed it without treatment or biogenic feed. And that one I show it in here. And this project actually funded by the United Nations Development Program and University of Malaysia, Sarawak. That time I worked there as a senior lecturer there. And that's the total fund. First time is the fund is USD 120,000. And the second time it's extended because UNDP asked me, you said you are rearing your brood using biogenic fish feed. We want to see that one. So that's why they extended another 8 months. That is up to June. That is October finished, then November to June 2023. That time I rear my mud crab, my brood crab using my lactosec. Then I breed. That time I get the good survivability. That one you can see here, and also less cannibalism. And this is the basic of the project. And project activity setting. When we get the project in October, our main intention is first year. First year this is, and that time I already mentioned we got in November 2021 and until October 2022. That time our plan activities of this one. Plan activities are we just had a survey, a feasibility study, then crab farm, design, construction, biogenic feed formulation, and the most important is biogenic feed formulation. I formulated in Malaysia and Malaysian local feed ingredients, but I didn't use the same ingredients for Bangladesh. I tried to use this feed formulation using Bangladesh local feed ingredients. So that's one. Biogenic fish, I just use some probiotic here. All take that one is a lactosec, that one. And then even stimulant preparation, and then crab breeding and sampling and analysis. These are the 1 year, and that was we didn't complete within 1 year. We just tested our constructed hatchery whether it works or not, so that we breed some natural. Just collect the gravit and then keep it in our pond, and then we just see the natural breeding. That one. And then the second year we just is a continue. This farm they will produce the local baby crab to the farmers for further brewing. That means fattening or soft shell, hard shell. They just making like that. And Bangladesh is once upon a time Bangladesh is the rich of prawn and shrimp production, but nowadays mud crab takes big space for that one. And this industry where we do, they are the number one industry in Bangladesh, that one. And activities, what we do in November 20th, this is 1 year. We first survey and feasibility study. That one we do many field visit, and you just see. I just give it some shortly. What we do salinity gate, because when you come from the mangrove and to the inside your household, that time salinity is less. Though, so we just measure the salinity where the stable salinity we get. We just select that area, and finally we select JFTL. That means Japan Fast Trade Limited. They are the number one mud crab production industry. This one. They produce soft shell. They produce hard shell. And then this is the construction layout. Or we do the AutoCAD design, and then more than 1 acre of the land. We asking Japan Fast Trade to give us this project more than 1 acre of land, and they already provided. Then we set up this one. But honestly, I just say if it is farmers level project, you don't need to water treatment or anything. Just you need only 1 pond, 1 or 2 ponds, and some ponds you just collect the brood from the nature. And the number 3 works is mud crab trial breeding. Trial breeding means I already mentioned when we do the pond excavating, we setting the design, that time consuming, and we lost the natural breeding time. And that's why we just take 1 gravit from the nature and we give it this gravit to our new constructed pond. And after a few times, that is month of the June. Bangladesh natural breeding time of the mud crab is starting from April, sometime mid-April, and until first week of July. This time they are usually natural breeding time. So we already lost, but we just take this one some natural breeding, because it's quite difficult. If we do at least when you do the biogenic feed, at least it needs 12 weeks. 12 weeks is the minimum to feed the mud crab for make it more sustain, that one. So this is the natural breeding, and then we can see you just see this one. You can see this one already some baby, the natural breeding. Because we take this challenge. I already said many of the scientists, many of the extension trainers, this is local mud crab not possible for breeding, so that we just use this local breed. Then we just trial this one using our pond, and then we make the biogenic fish feed. Already I just say I use the local feed ingredients. And these local feed ingredients, you just see this all from, because my one student, Mr. Jahidul Hassan, he is going to submit his PhD thesis soon. So he actually helped me to what are the local, because he works in a fish quality control lab. and government quality control lab. So he knows what the local ingredients are available in that project area. So he just collection, and then when I make this, then many persons say it's not eat. Crab usually like to eat the nature food. So is pellet food they not eat. But we success this video, we make it in next slide, the bigger, then you can see the crab already take my formulated feed. Then even distributing prepared feed, the my another student just now he already got the doctor. Now he will going to present his findings, some comparison, and he actually take care the brood and all time brood checking, and then grab it to brood. When he's grab it to brood, that is he checking the color of the egg, and then because when we do some project work, that should be more systematic, so that we see the color, and then he monitoring all the time and feeding the feed quantity, and feed quantity, if it is more, he just give the crab is color is changing or not. Then he just, when his color, he just take it in the pot, only 4 crab. And this is he sometime do the verification really what feed we give, it have a probiotic or not. You know, probiotic means is a live microorganism. Here is 2 probiotic. One is Saccharomyces cerevisiae, this is live yeast. Another one is Lactobacillus acidophilus, this is bacteria. Another one also have Enterococcus faecium. Enterococcus faecium actually not a probiotic, but it's a sensory microorganism. Sensory microorganism, it creates some smell to attract the aquatic animals to take that food. That is, so this is have or not. He just tested in periodically, and then he keep it because of his PhD. Also, he needs to really say the probiotic feed have or not. And then sampling analysis. This is when this breed. I say already 50%. We just collected. We just keep it in the laboratory test. Then what happened? Why conventional hatchery produce less survival rate? That's one. We just tested it in the laboratory analysis, and then say during JUA time, during JUA 1, JUA 2, JUA 3, JUA 4, what is the reason? So this is laboratory analysis we do, and then after that, when he get the feed, after 28 days, then he just collect that one. And this is the second phase. That is, we use the biogenic fish feed. That is completely follow my project design. That means we rearing the gravid mud crab using formulated biogenic feed. And then you just see one, we just write down here, the hatching zone was pin drop silent, covered the container with black garments with gentle aeration. That is, this one we found it during the natural breeding ground. Natural breeding ground where they're breeding. The farmers also say that time they usually not plying the boat. The temperature is when they have the practical experience. Now they are going to breed. So that's one, they're not disturbing that. So we also maintain the same environment to our hatching land, our hatchery place. This is natural hatchery place. It's not any concrete building. It's like a pond. We just give some silent zone. Everybody not permitted to enter the hatchery, the earthen pond area. Just we ensure this area should be pin drop silent. That one. And then when it's hatching, we give live feed. And live feed also, I just tell you something, don't give copy pot. Don't give copy pot, because copy pot sometimes is bigger than the joa. If it is joa, because copy pot also the cannibalistic. So if it is bigger live food, they also eat the smaller joa. That's when the survival rate is less. So without copy pot, you can use rotifer. Rotifer we culture, and we give this rotifer to the JOA 1, JOA 2. We also give here, I will show you that what level, what live food we keep. This is the second phase. That is, I just say you when we start the biogenic feed. That is first November 20, October first phase finished, October 22, and then November this is my PhD student Abdul Hannan. He recently get the PhD from University of Malaysia Sarawak and also awarded. He he actually do it here for identification of the mud crab. What is it? Scylla celata or Scylla olivicea or other two have or not? But he found out it's the Scylla olivicea, and this is our brood rearing pond. Brood product. You just see this is when it's in dry condition. First, you make the dry and then free from all infection. Especially you just fully dry and then you just keep it. And then this is, you can asking this is some polystyrene because Bangladesh this time November, December, this is the winter season, November, December, January. So we not give any of the artificial thing. We just use this polystyrene. Just polystyrene in the sunlight time, daytime it's hot. This is hot, and night time the temperature not down because of it's already some sunlight energy. That's when water temperature is optimum, should not be dropped below 20. If below 20, it is in Bangladesh, but in Malaysia, Thailand, I don't think so. It's have a problem because this area have a no winter season. Also, rain time temperature should not be down below 20. Still temperature, water temperature is okay. But in Bangladesh, this problem, this is the winter season, so we just keep it this one. And someone just say me, sometimes we give the aeration, then fully I just say, okay, aeration also, sometimes aeration not fit to maintain the temperature. So this one, we just keep it, the temperature time, and here down lot of brood. One is one brood. So the temperature, all is, we just give them fully environment, what usually they get from the nature. And this is survey, breeding ground survey. And my two students also here, they also all time regularly visiting the ground, natural ground, and then back to the our project area. Just see, just what I say in computer and troubleshooting. They also see there and then come here. If anything have this time, they are take it very seriously. Very seriously. And the most important is this time is the COVID time. But Bangladesh actually not too much restriction. So that I didn't also go during this time to frequently not go to the project area. But all time, this 1 year, 1.5 year, actually, I 24 hours, 7 days, I fully communicate with them. I just take their video. And then I also disturb these two guys. And another failure, what about the result? That one. And this is after rearing, the egg is like this. And this one I already show in hatching all they started. And this is the crablet. Crablet appear after 26 days. And if you see this, there are some crablet already. These are it's more clear next year, and then we produce our natural crablet to the UNDP beneficial. That one, this one. So this is crablet appear, and this is hatching already. I show it before, and then this is that box, bamboo box. This is a spawning box, which is a bamboo made a spawning box. It has been made followed by the local box, bamboo box from this, but this mesh size is we maintain the mud crab brood cannot be out from this box. And this is also as a patent, my 3 IP product, one is this one, and I use the local materials and then we make it. This is the natural. This is the drawing, this is the natural, and then inside there is a food, biogenic feed. And when I went there, I also help my PhD students, all are setting. If you see this, all are setting one bamboo box, one mud crab, granite mud crab. Then we give the feed, and I actually randomly checking. My PhD students just say every bamboo box have a mud crab and give the feed, so that I just randomly checking really mud crab have or not, and give the feed or not, so that I just when I visited, I just see this one. So these are the thing, broodage development, and this is the live feed. Live feed already Jua, we only give microalgae and rotifer. Jua 2, we give microalgae and rotifer. Jua 3, microalgae, rotifer, and artemia. I already said don't give copy pot. rotifer is good for mud crab, Joa and early stage development. Joa 4 will give rotifer and artemia. Then when it's Joa 5, we're just counting the day. Honestly, we not see there where they are Joa 2, when Joa 3. We just counting the time. We just change the feed. Microalgae we test. We give rotifer just when give microalgae and rotifer just sometimes equal percentage of rotifer to equal percentage of microalgae. Then some microalgae rotifer artemia, we give a small amount of microalgae. Microalgae is good for rotifer. Rotifer eat this, and rotifer is fed by the baby mud crab. And then artemia. Then when it's a big size, artemia we produce. Joa 4 also is like a copy pot size, but we didn't give the copy pot because copy pot is also active carnivorous. So that's when we just give it rotifer artemia, and Joa 5 we give artemia. Megalopa we also give artemia. Crab instead we give artemia. Then when crab instead we also introduced our biogenic feed, very small amount powder form. We just give it to the pond, but nursery zone. That one. And then this is the culture method. What a microalgae. This is the very much scientific. So that I don't think so. It's necessary to the farmers level. It's only if you do some like this project, that time you need to how to culture, because it must show to the when Hannan do the PhD, he show to the examiner, yes, this is microalgae, this one. And this is the dietary biogenic fish feed. Dietary lecture, we maintain the feed formulation 45% protein, 6% lipid for breed stock management. And dietary lactosec feed formulation 45% protein, 6%, and then we only buy lactosec from Alltech. Others all ingredients for protein source, for lipid source, lipid, everything we bought from we use the local materials, local feed ingredients. That means fish meal also, this is local. Fish meal, if you use the local fish meal, this is protein content usually 56 to 58%. So we just making the formulation, we give five types of formulation, 5 types of feed, and then which contain 45% protein and 6% lipid. That one we selected. Usually carnivlastic fish species, they require lot of protein and lot of lipid because it needs energy. So if you give less protein, less lipid, you cannot success. So that this one, I my PhD time, when I do my PhD, I also do Asian snakehead. Asian snake is a passive carnivorous system. That time I maintain 40% protein, 12% lipid. But here, when I see, this is just my assumption, and I see some literature reviews, some Indian scientists, they also use 45% protein and lipid. So that I just maintain this one. And then all local materials, and then added only 0.1% lactosec. That means 1 kg, you give 1 gram lactosec. Mixing with this, and then mixing the full feed ingredients in a homogeneically. You just see when you mixing, when you see the color of the mixing is homogeneous, that time you're thinking that your feed is good now. If color is some different, that means it's still your protein, lipid, some portion have, some portion no have. That's one, you just make it, and then make it by using a mixture, as well as some meat mixture. If you want to make some pellet, you can use some meat mixture. Or if you don't use the meat mixture, you just make it, the mixing, just make it in a mold, just like when you make the bread or roti. That time before making roti, you make some mold. That one you just make that small, you give it to the brood mud crab. Automatically, your brood mud crab is already habituated to eat this one. And the most important is, it's a probiotic. Probiotic, what helps? Probiotic helps the digestion more. Digestion enhancement. And that one also found my PhD student, because when he do the PhD, he do A to Z analysis. And he found he did gut microflora. He conducted what are the development of reproductive organ, what the development of the hemocoel, what about the immunohistominate, what action, everything he find out. So that's one, when you give the probiotic feed, it mixes it enhance the digestion. If digestion good, every biological cycle will be good. That's the philosophy of the lactosec feed. And this is the habituated to formulated feed. If you see, I want to show you this video. The previous video is a very small. You can see how our brood mud crab, gravid mud crab, they eat our feed. This is number 2 days. After 2 days, because we give feed, but the crab not take interest to take. But I just ask them, just wait. Within 2 days, you just wait until 4 days. If you not eat, then we can change the formulation. But Alhamdulillah, the mud crab start to eat after 2 days. 2 days, that one. And this one also very silent mode in the night time. If you see in the night time, you see now they moving their ear, like how they eat that one. Because when Enterococcus faecium mixes with the probiotic feed, it produces some smell, attractive smell to take the feed from the. You see, already eat. You see, and you cannot give more video picture because I say it should be pinpoint and don't disturb the gravid mud crab. But my field staff, that is, he is now in Italy. He is too much curious. He's too much curious how mud crab eat. So he just take the torch and then take the video. This one. This one. You see already fillet they are taking using their finger. I think everybody you watch this one, right?
Yes, we can see it. Dr. Munir.
Yes. So we also tested the cannibalism rate of crablet from the natural breeding. I say we didn't use any antibiotic. We didn't use any chemicals. We only use probiotic. Probiotic feed. When the baby crablet come, we just take 1,000 crablet from the natural ground, our pond, from the pond, and then we just take 3 replicate, tank 1, tank 2, tank. These are the concrete tank. Because where we established our hatchery, they have also conventional hatchery. So we want to see what about their survival rate, cannibalism rate, because it's not a stress. Without stress, they also eat. Their active cannibalism, they also eat. So we found around 28.8% survival, 29.8%, 27.8%. Here we didn't use any biogenic feed. We use here just freedom. We just give tilapia flesh. Tilapia flesh we give to the newborn crab and they eat. But sometimes when they are active cannibalism, if they're not hungry, they also eat each other. This is called active. And passive difference, if they are hungry, they attack each other. If they're belly full, they never attack each other. This is called passive carnivorous. But active carnivorous, whether they eat, whether they're hungry, whether they're not hungry, they attack each other. And carnivorous mud crab is like that. So this one, they are stressless baby mud crab. What about their result? That one, we get a very nice result, and this is not a very much significant difference. One tank one, we get survivability 28.8%. This one, we rear 60 days. 60 days we rear. And 28.8 days, we get our one crab is 80 gram. 80 gram one year. One year. Then when they are eating each other, you see there are one piece of the mud crab, how they're cannibalistic. My PhD student, he just collected some dead mud crab in there. And then survivability 28.8%, 20. Then after that, this 80 gram, we release it in the pond for either soft shell or hard shell by using that particular farm, that one. This one, we transportation our crablet to the beneficiaries. What we do, how we transport that one. You see, the mud crab is so much fast. Have you seen? And I also show the comparison. What about the conventional hatchery, crablet, and what about our hatchery? And this is our fourth cycle because UNDP give me target at least fourth cycle. We must do it intensively. The cycle one, that means one crab. One crab, how much we get total jua, others we counting. This is the estimated actually. Jua, because we cannot count the jua in a physically because it's quite difficult. So we just estimate, just wait and follow the literature review, actually what they're doing, and according to the weight, our maximum weight of the gravid mud crab is 130 gram to 140 gram. So 130 gram to 40 gram, whatever the estimated egg usually when the season, these are we just counting, and then hatching rate, what we do 85, and the hatching rate we counting because we waiting 2 hours or sometimes 3 hours, just waiting until they are hatching. So if they're complete, they don't have any egg. That time we just sure this is already 100% or 80% already hatching complete. Then crablet, when we after 28 days, that one when we get 58,774, and then when we calculation, the survival rate is 12.58. 12.58. That means average survival rate is 12.58. Here I mention actually we also learning because nobody can adapt this technique. We also use that time. We also make some hapa. Hapa means some small net in the harvesting place. But mud crab is a highly carnivorous, and they just broken that net and then left. And then when it's in the soil environment, this is mud crab. So they all time bait. They just pants the muddy zone. Soft zone they just go from the muddy to other places. Overall, all these challenge we get this number 12.58% is a good compared to the better than the conventional hatchery. Conventional hatchery you get maximum 3.2% if you use control environment. Sometimes if it is not controlled, the commercial, you cannot get more than 2%. More than 2%. So this is one of the good result and significant result, 12.58%. And then what we learned from this project succeed to use local mud crabs for reproduction, and the scientific name is Scylla olivicea. This one we find out. And in natural mud crab, you don't require eye ablation. If you ask me why not, we also did eye ablation. With 12 gravid mud crab, we also do the eye ablation. Eye ablation helps to early maturation. But in this stage, when we do eye ablation, our all gravid mud crab die. All mud crab die because of the close environment. Something maybe when eye ablation, we not give them, because when eye ablation, you already insist to breed. So that's why maybe mud crab don't like this, and then no need eye ablation. You just rear the gravid mud crab in the box. Just normal that you take the patience when they're coming for the mature. Then without antibiotic, without chemicals, you can use this one is a natural. Hapa is not, although we give the hapa, but hapa during mud crab reproduction, this one sometimes is broken, that one. And if it is near the cyclone zone or others, also natural disaster also have a problem. Cock sheet, this is useful for our Bangladesh because of the winter season to maintain the temperature. I not give any chemicals, anything, so that I use cock sheet, and that cock sheet helps me to water temperature stable. Harvesting method, we face lot of difficulties. We use one method from the local farmers, feed and trap method. That means we give some bamboo and put it some tilapia mussel, and then during full moon, that is the nature time, when we give it there, the crablet is coming and that's eating that one. When it's eat, it automatically catch. That way we collected the harvesting. But harvesting, we just suggest that harvesting is one of the difficult tasks. This natural breeding, we must do some more concentration to harvesting. I think local farmers, they can they can do some their local knowledge, but we success 12.59%, we using only feed and trap method, this one. And our biogenic dose is 1.5% is the suitable. And for antibiotic test, we do it in government lab, Fishery Inspection Quality Control Lab, Department of Fisheries, Bangladesh. They not find out any antibiotic of our produced carblet. And this is one of the good result. Now I show you, I already say where we implemented the project, they have also conventional hatchery. They also produce crablet from the conventional hatchery. If you see this crablet, this is conventional hatchery produced. The crablet not moving. You see, crablet is live, but they are produced from the conventional hatchery. The crablet not moving. They are very much stressed. If you see this, when you give the water, also it's not moved. But when you transfer it to the earthen pond for further processing, for hardening or soft shell, because from the beginning it's a stressful baby mud crab. That's why it's not sustained, and then the survivability is less. If you see our one, we already say we make it in a hapa. This is the hapa, and this is one of our local worker in the project area. And the tone, the tone is my PhD student. He just taking. You
আমরা
just see.
আমাদের এই হাপা থেকে ক্রাবলেট হারভেস্ট করছি
He
এখানে
say in
আসলে
Bangla.
মামুন সাহেব
He's the person. The worker name is Mamun. He just taking our production
দেখি আসলে
crablet.
ক্রাবলেটগুলো
You
কি
see.
পরিমাণ আছে
You see crablet. They are moving
প্রচুর
without water. They are moving and they are very much healthy. Very much healthy. This one.
একটা টানেই আসলে আমরা এই পরিমাণ ক্রাবলেট পেয়েছি
This is our success of this is the natural mud crab and local breeding. And so what is last year, the model of sustainable natural mangrove based mud crab hatchery is 100% alternation of the conventional hatchery for production of the mud crab. It means actually if you really want to do crab production, no need to concrete based hatchery. If you have a pond, you can set up, and for pond salinity 26 ppt, you can set up, you can establish the crab breeding in your pond just making a bamboo, because it's a active cannibalistic. To protect from the cannibalism, you just introduce that box and the rearing the mud crab, gravid mud crab using this biogenic feed. And there is no need separate conventional hatchery for mud crab. Already I say, if there is a community level, you can select some area, then you produce the mud crab this method. And the last three is, this is very much now world, this is one of the most attractive, more attention, 3 zeros: zero poverty lead to zero hunger, zero employment, and zero emission of carbon. These all attributes available in this natural mangrove based mud crab hatchery. Zero poverty, if you have a single pond, you can just collect from the gravid, from the nature, rear it in your pond, automatically it's breeding, and then you collect it after 28 days, then you just keep it within 60 days, you can produce mud crab to the industry person. That's the name is, you can sell this, and then if the survivability is more than 12%, a crab usually they breed more than 100,000 for Scylla olivicea, but if you do Scylla serrata, it's a 1 million. So and Scylla serrata is less cannibalistic compared to the others. So that's why if you success this in your pond, your poverty level will be less, and you will be one of every trillionaire person. And then you also create some young generation for employment. That means zero unemployment also created. And here there is, you never use antibiotic, never use any chemical, and so it's a zero emission of carbon. So this project support all 3 zeros. And this is the more information if you want to know. We have published another paper also going on in Aquaculture Journal.
This one our already published. This one published by my PhD student. If you need more, or if you contact with me, I can proceed you. I can send you this one. And this is our animation. How our hatchery is going on. See this? Before when you do the design, that time we do various types of troubleshooting. We just drawing AutoCAD. Then we make this video, audio here. This video, this animation, we just make it. But for farmers level, no need water treatment plan. If farmers level, if you water treatment plan, you only set up the sand filter, sand water. That is low cost. But here I just say this is the project. Also, it included the PhD degree, so that it's need more sensitive, so that we use this thing. But in farmers level, no need of your connection to the natural, to the your area. You just take the water from there, and the salinity increase. If salinity is 10 to 12 ppt, you just store some water. Then after few days, it automatically, when it's dry, automatically salinity is more. Then that one you just maintain. If it is more, you just reduce. If it is high, you just mixing water. That one. Then you maintain 25 to 26 ppt salinity. You just see, we make it the box. And same model we use. And you see some black polythene, because we maintain the natural environment in the pond. This is the black polythene. Black polythene, we just give it. Similar design. If you go in Bangladesh, Japan fast trade, you can find this design in that area. They are also going to produce this mud crab from the natural area. I just wait until ending this animation. The design is also my intellectual property, also registered in Malaysia, international. This one also copyright the design. And bamboo box is a pattern. And biogenic fish feed actually is before. Biogenic fish feed, I tested 9 fishes. Also, Sarawa, one of the most sensitive fish, Ampurao, is a slow grower. But biogenic fish enhance the growth performance also. It's a very much low cost, and UNDP, we really acknowledge to UNDP, and UNDP give us this opportunity to do this work in Bangladesh. And this is designed by me and my 2 PhD students, Abdul Hannan and Jahidul Hassan. Samsura is my Dean of PBS. And this is my IPO copyright. And that's end of my presentation. If you need further query, this is my address. I am working in
University Islam Sultan Sharif Ali as a senior assistant professor there. And my intention is to sustainable aquaculture. I don't, for if you do some reproduction work, don't adapt the modern technique. Modern technique you can do for faster growth. But if you relevant to the reproduction, kindly adapt the natural system, sustainable way. That's all. So recently, I already say my PhD student already complete. He wants to present after my presentation. He wants to share his presentation. He see the comparison of natural breeding, nature. He just reared the brood in the natural, then bring it to the conventional hatchery. What about the comparison? That one he is going to present is also nice findings. So I would like to request to Abdul Hanan to share your presentation. Abdul Hanan, are you ready?
Yes, Prof.
Okay, so you can present.
Okay, thank you for giving me the opportunity to present a comparison about my research study. And good afternoon, Assalamu alaikum. Actually, I would like to show some comparison of our conventional breeding and mangrove-based natural mud crab breeding. First of all, we know that there are two methods that are actually applied in mud crab breeding. First of all, the conventional breeding that is very familiar to all over the world, and this is the very new technique that is nature-based mud crab breeding. It's not actually new; it's very popular and very common in nature, but new to for our human. Actually, these two methods are different in terms of ecological impact, productivity, sustainability, and mud crab aquaculture. Actually, this is a very useful and sustainable technology for mud crab breeding. And I hope in future it will lead the breeding technology in all over the world, this nature-based mud crab breeding technology. And there are some comparison. First of all, the conventional hatchery is concrete structure that is need high investment cost. Actually, the concrete structure mud crab hatchery is not possible to for all the people living in middle class or lower class. Actually, this is the intensive and high intensive cost structure. But in case of natural breeding technology, this is a very small structure and low cost structure, and which easy to handle this technology. In case of conventional breeding, the broods are collected from the deep sea or the lower part of our Cox's Bazar and Chittagong region. But in case of local brood, in case of mangrove-based technique, we collect just local brood. Actually, it's very feasible to collect, and there is no need to transportation cost. And actually, in case of conventional breeding, when we collect broods from the longest area, Chittagong, that's very difficult time for the brood. But at that time, brood are very sensitive, and they need to transport around more than 12 hours, and that reduce the immunity of the broods. It's very destroy the quality of the brood actually. But in case of local brood, there is no transportation cost and very easy to collect. And the main basic thing is that we collect from the natural environment, and we also place the brood in the same environment like this. Another point of conventional breeding is that the poor immunity and high mortality rate due to this long transportation. I already mentioned that. But in case of a local brood, they have a strong immunity because they fight, they struggle in natural environment, and they have the strong immunity, and the mortality is very lower in case of local broods. Another problem of conventional hatchery is the uses of antibiotics. Actually, antibiotics and groups of drugs are used in conventional hatchery. Uses of antibiotics already Prof mentioned that there's reduce the immunity and make create stress, and also this is not feasible for the environment because of I'll go there. But in case of natural breeding technique, there is no uses of antibiotic, no uses of hormones and like this. Meaning that the uses without using of antibiotics improve or the immunity of the broods. And another important thing of conventional breeding is isotope ablation. And there is no isotope ablation occurred in our natural breeding technique. Actually, we breed crab naturally in our technique. Here is the effect of isotope ablation and conventional breeding. First of all, stress and mortality, because sometimes in hatchery, the isotope ablation are conducted in single or both eyes that create stress and sometimes mortality due to infection. A lots of pathogen like virus, bacteria, fungus, parasite are infected this infected region and occurs in mortality. And another thing is reduce the lifespan. The isotope ablated brood stock have reduced lifespan. That is already mentioned in different literatures. And the egg production are decreased, and this with poor immunity when we created the broods through isotope ablation. And the effect of antibiotics, already my professor mentioned that the effect of antibiotics in animals as well as the humans. And the uses of broad-spectrum antibiotics is very harmful for the animal and even our mud crab breeding. Because of we didn't consider, we just consider our production, but we didn't consider our immunity. Immunity is the main factor for the for production of mud crab and the survival rate. Poor immunity poor immunity of brood produce poor quality of crablets, and the pathogen are present in crablets. The use of antibiotic, they make resistant to the pathogens. And another important thing is that the effluent discharge from the native environment, native river, native gate, and that is the very dangerous for our environment. So it's time to avoid our antibiotics in our environment. And in case of natural breeding technique, there is no stress, there is no egg production, poor egg production, or immature egg production, because the egg are hatch or egg are spawn after full maturity of the broods. And a strong immunity of broods produce a strong immune crablets. Another important thing is that this is the sustainable technology. There is no carbon emission occurs in this technology. And this video already shows professor that this is the species produced in conventional hatchery. There's poor immunity, that less movement, but in case of this, I would like to show this video also. This active movement of the crablets. This produces a strong immunity. They struggle in nature, they sustain in nature with a strongest and immunity. So these are the reference I observed in this these two technology. And this is all. And thank you for my presentation and attention. Thank you.
Thank you very much, Dr. Abdul Hannan.
Thank you. Thank you.
Thank you, everyone. I think we still have a lot of participants are still continuing. So as I said, now it is the time for question and answer. So I will kind of keep the floor now open for some times, and I will try to see if we have any questions in the Q&A box or in the chat box. First of all, thank you very much, Professor Dr. Badrul Munir and Dr. Abdul Hannan for the wonderful presentation that you have made. And indeed, it was not only very informative, but also I found it very useful. And I believe all of our participants have found it very useful and very details. I was thinking like, where is actually the area we can ask some question? Because literally you have already answered all the questions that we might have. So let's see. It is actually part of our webinar. We need to see the Q&A. And I see there was one question, I think, in the Q&A box which has already answered privately by Dr. Abdul Hanan. The question was from an anonymous participant, which is about the breeding season in Sundarbans. So I understand the breeding season in Sundarbans means they're talking about the breeding season in Sundarban part of Bangladesh. And the question was, is it January, February, March, April, or October, November, based on different research? In Sundarban, mud crab harvesting is banned by Bangladesh Fisheries Department during January, February, as they claim the season as the peak breeding season. So Dr. Abdul Hanan has already answered. Actually, it is very confusing in literature. Mud crab breeding started in March and continued up to August or September. But peak breeding season, I found, Dr. Abdul Hanan said, I found in April to June. Okay. Now, Mr. Enamul Haq is asking, is there more published paper on this? Other than the paper published by Professor Rauf and Ali in 2020 and Islam from JSTU, Professor Islam from JSTU. So, Dr. Munir, would you like to add anything? What is the real breeding season for the Sundarban part of Bangladesh? Do we have a clear information on that?
Actually, we didn't actually perform in the open water where they're breeding, but we only gathered the information for the local farmers, because before establishing this natural mangrove hatchery, we collect the information from the local farmers. Also, I mentioned it, we also visited the local breeding ground. And local breeding ground, yes, January, February, maybe that time ban of the Bangladesh government because of they ensured the pinpoint, because there is silent environment needed. Silent environment needed. I all time mention is needed. Also, farmers also say the mud crab natural breeding, when it's start before few months before, is completely silent. If silent, if not ensured, they are not breeding. They are disturbing their biological cycle, not breeding the system. So actually, this study not find out what the actual breeding season, but in March, April also, when our rearing gravid, we also waited long time when it's come, because we depends on the nature. We depends on nature. That time we also frequently asking the farmers who actually collected the natural crablet from the river. Did they see the crablet or not? They also not see the crablet, and that's why we just assume that when our first breed occur in April 18, if I'm not mistaken, first hatching 18. Hanan, is it 18 April, right?
Yes, Prof.
18 April. 18 April, our first hatching. And then after 18 April, it's sequentially, actually here we show it four cycle, but actually we breeded around 12 cycle. And continuously it's breeding until May. And then when May finish, then again takes time. Then some very occasionally, just one or two they're breeding. That time our project already, our commitment is we only give the fourth cycle. Then we already complete. We just give it to the project industry person to rear it. So our time we already success for 12 or 13 cycle, but we only produce here only 4 cycle. But survivability all is like that, 12.49, sometime 11. So that we just conclude our thinking is breeding season might be April to until first week of July. But mud crab actually breeding surround the year. If they get the environment, they breed all the time according to the literature.
Because we have participants from different country, I just want to make this clarification. So these months or the breeding months can be different for different country, correct?
Yes.
So for Thailand, for Malaysia, it could be different months.
Malaysia, Thailand is a tropical region. So it's a tropical region, temperature all time 26, 28 is all time maintained, and then it's I think it's around the year. Maybe some is the peak season, but all the year it's a breeding. It's a breeding, but when you do the culture, I just mentioned you what I produce that feed. If you want to know the formulation biogenic fish feed, it enhance, it stimulated the breeding of aquatic animals. So that's why if rearing this, it's a breeding surround the year.
Thank you. Thank you, Dr. Munir. So I just wanted to check because I think I got the information, but not sure if you have also shared it. Like, what is the survival rate in the nature? Do we have any data from the nature when they
No,
complete in the nature?
no.
Okay.
We don't have any survival rate because the study Handan, you have any data of the nature?
Because in your
actually,
technique, it is
it's
up to
very
12.
difficult and it's very
Very difficult.
yes,
Very
to
difficult.
say, because we just make an study which is simple part of the nature.
Because in your presentation, I got it like it might be lower than 1% or less than 1% to 7.
That is conventional hatchery.
Yeah,
Conventional
that is
hatchery.
conventional, but you've got 12%, correct? 12%.
12%. My own is 12%.
Okay. Now there
And
are
for your information, because we adapted this technique in a industry. That industry actually one of the partner of the JICA. JICA, Japan, their scientists also visited our hatchery. And they just like this presentation, they just acquire all the knowledge and they adapt it in their country. In some small scale, and their survivability, they inform us around 16 and a half percent.
Which country are
Japan.
Okay.
Japan, and they also say 16.5, but they just informed to my PhD student, you get 12.5%, but I get more than 16% survivability. So that's why they are the more what we face difficulty. Maybe we also expect our harvesting, because some mud crab harvesting is one of the difficult tasks. So if harvesting 100%, it would be maybe it's not a 12.5%, it would be more survivability. Because after 2 months, 3 months also we get many crabs, many mud crabs in the pond. So we cannot catch them during the harvesting. Okay,
great. That's a great information. So is this the same genus, the Scylla in Japan?
Yes. Scylla olivicea.
Okay. Now there are two questions that has come. One is from another anonymous participant. How to prevent copypod in the culture pond? Because you said not feed the copypod, correct?
Yes, this is one of the difficult tasks, because when we prepare our pond, we actually fully dry. Number one, fully dry, and we use tea seed cake. We use tea seed cake because we use all organic. We not use any inorganic that thing. And then after when water refill, we also test it. Copypod have or not.
Okay.
We didn't find a single copypod, and so that we sure this is the way. This is the way of copypod reduction. And I just request all of the practitioner, don't use copypod, don't give chance to copypod near your pond area, because copypod is very dangerous for mud crab production.
Okay. Now let's say some of the farmers who have already started this pond and the and the breeding, and now in case if they need water to be added, what is the process that you are suggesting? Because you were saying that
is it saline water? Saline water, or
yeah, I mean they might need to add water because the water is evaporated or water is leaked. So then if they need more water and they want to bring the natural water from natural water bodies which might have the copypod, is there any suggestion, or we need to go for treatment of that water before we?
Actually, when we our natural mangrove hatchery, we also established a reservoir. A reservoir of the saline water that is directly connected from the river. And then so that I just say, if like that you just establish some sand filter. Sand filter is not a costly. Sand filter costly, but here we use also water treatment something. Then we just store the water from the river.
Mhm.
And then water river, the salinity is around 10 to 12 ppt, which is not a 26 ppt.
Okay.
That's why when it's stored in the reserve,
Mm.
then once upon a time it's increased.
Mm.
And then this is the project so that we also use sea salt.
Mhm.
Sea salt, we just buy sea salt, we just mixing with the water body. If salinity drop, we just maintain 26 ppt, 25 to 27 ppt we maintain our water here, and then we use sea salt. And because this is the project, we have also three sources. One is reservoir. Reservoir is very much common for the farmers who are the practitioner. This is no need huge money, just store the natural water, then once upon a time it's a salinity. And if it is community level that have some project work, then you can buy the sea salt and brine water, that one. Because if it is community level, that means have a project, you just make a one big hatchery, you can introduce this thing.
Okay. Thank you very much, Dr. Munir. I think there is one more question that said, if your technology commercially viable to produce enough mud crab to replace the wild catch, is your technology commercially viable to produce enough mud crab to replace the wild catch? This is the first question.
Hmm.
I mean-
Can you repeat again?
Do you think that your technology can be the replacement for the
Yes, it's
traditional
100%. 100%
traditional
replacement.
practice where the farmers are actually bringing the baby crabs from the nature?
Yes, this is because baby crab of the nature, some countries different rules adopted. Usually, if we save our biodiversity, then all countries they have some rules and regulation to stop the catching the baby crabs. That does not lose the biodiversity. But this is completely natural. Completely natural. We just introduce same technique what usually adapt in the nature. Maybe we not provide them like a because mud crab, when they go for the natural breeding, they usually breeding time they go very deep water. Very deep water, dark condition. That one we not introduce, but we introduce this thing just artificially dark environment. Dark environment, that means we can give some black polythene of that one, and that one we just introduce this one. And also this is if it is like this, it's a good for alternative of the natural or wild baby mud crab alternative.
Okay. Yeah, thank you. I think I got it. But the person who is asking, his name is KF Iftekharul Alom. Probably he is asking the question in terms of the supply and demand, because his second part of the question is, the GCA project of UNDP did not obtain necessary mud crab from your project because of the shortage of supply. Can you explain why?
Okay. If it is UNDP person, I think it will. I explain. I answer this question, but also I asking Doctor. He is a new doctor, Abdul Hanan. He will answer because that time I was not in there in the when they are supply. The person who in charge UNDP, he just taking lot of tablet. My Abdul Hanan, my student, also that time he was a student. He also say don't, because tablet is a highly cannibalistic. Don't store, don't, when you transfer the polyethylene box, it's not be high density. But he take that one and he carry this completely unhealthy condition. That was not when Abdul Hanan advised them, they not follow this instruction. And
Okay.
then they actually give us
We got it. Maybe because it was kind of a management
unhealthy
or
harvesting.
handling issue, I would say. But primarily, the question is, let's say it is the primary question that we have been asking, or Mr. Iftekharul has been asking. So let's say if your technology or if your method is really a viable method for the local context, for the local farmers to easily adopt this method, do you think we can replace the traditional practice of catching the wild baby mud crab from the nature? Let's say
I
your
think-
food is widely being
Okay,
practiced.
okay. If we produce this one, I think we don't need to catch wild from the nature.
Okay.
Wild nature, and the number of mudcraft. If Mr. Ritika say like this, because we only do 4 cycle.
Hmm.
4 cycle, and our contract is 4 cycle only we do, and 4 cycle we get. This is accurate estimation of our production, and that time we got 12.59, that one. But if we do really is a large scale, that time we can get, I already say, we suffered the problem during harvesting. During harvesting time, also that area affected cyclone. During UNDP person, you also know 2 cyclone attack that time in mangrove area. So we also lost some of the mudcrab, baby mudcrab. That's why we not get. But when we do intensively 4 cycle, that one is the actual result. So if we take proper attention, the attention of that, we must do some natural breeding, and we must save our biodiversity of mudcrab. We can easily adapt this technique, and it's
Thank you.
very easy for the farmers level.
Thank you, Dr. Munir. I think with that, I don't see any other question in the Q&A box, but we have kind of a, not question, but it's rather compliments in the chat box. I see there are people like Dr. Christian Labbi from the University of Environment and Sustainable Development, Ghana, who is actually seeking for collaboration. I think we will share this information with you.
Okay.
Collaboration on shellfish breeding. And
Okay.
person has actually shared his email, Dr. Christian Larbi. Then we have Masood Rana, who has another compliment saying that it's a great achievement. Hopefully it will significantly contribute to the crab hatchery for the economic development of coastal farmers. That is from Masood Rana. Thank you very much. Mr. Enamul Haq. I think he's Dr. Enamul Haq, who is the ex-director of BFRI Bangladesh, now working with Bangladesh Forest Department and IDE on mud crab resource assessment, breeding and season optimization, and breeding potentialities in the hatchery in Sundarbans. Okay. So Dr. Enamul Haq has given a long message that in Bangladesh, we are successful in mud crab, Scylla olivacea breeding by BFRI, DOF and few private hatcheries, but those are not sustainable due to some technical problems like salinity, water quality, live feed, etc. At present, JICA initiated hatchery with private sector in the bank of Sundarban. Breeding are more successful in Cox's Bazar region. In Bangladesh, majority is Scylla olivacea. I am interested with the present topic of natural breeding of mud crab. So if you like to say anything about his interest and his knowledge and information.
Yes, actually, I also thank you for BFRI. Actually, this project also with the help of BFRI. Honestly, I just say it's a help of the BFRI. Because my my PhD student, although they are the student of the UNIMAS, University Malaysia Sarawak, but they didn't get any facility, laboratory facility from UNIMAS. They get help from the BFRI, and BFRI actually tested this result. So that's why this is also, I just give the, this is also the product of the BFRI. Although this project is UNIMAS and UNDP, but also one of the owner of the BFRI.
Thank you. I'm not sure if Dr. Enamul Haq is still here, but thank you very much, sir. But one point that I like to bring here from his message, he said the hatchery or the successful breeding are happening mostly in the Chittagong side, but not in the Sundarban, which is Khulna and the Satkhira side. So do you think that is a correct information, and do you think that can be solved or not?
Yes, it's the correct information. But nowadays, Japan First State, their area is Hannan. Their area name is where? What?
Which one? Japan JICA project?
Japan First State. Their area name. I forget the area name.
Nildumur.
Nildumur. There are now after our project, there are some of the entrepreneurs, they already started. They already started because it's not a money matter. They have a pond. They started this work, and they take the box. This bamboo box. Actually, we make it through the local farmers. So they are trying. I think within 2-3 years, you will get some result from Khulna. Coxbazar, yes, it's right. Coxbazar, there are a lot of conventional hatchery, lots of conventional hatchery, but not nature hatchery. But this one, just we introduced 2023. Hopefully, within 2-3 years, if more people come, you will know it more popular. This method.
Thank
will
you.
be.
Thank you very much, Professor Dr. Badrul Munir. I think we need to end this today. It has been a wonderful deliberation from your side, along with Dr. Abdul Hannan. And I think we have learned quite a lot about the topic and also went a bit details to learn about the technical side of it and the problems and the issues, the pros and cons, everything. We see there is a huge prospect of this, and as we have talked at the beginning, the crab farming is really something that can be actually developed further and much more in all these countries which are having the coastal areas, especially the tropical and subtropical countries. But for that, we definitely need to develop the hatchery section or the hatchery part of it. And that is something probably your contribution or your deliberation was really, really highly regarded. I think we like to see this should be further developed. And then also, if there are also some collaborative opportunities that you can do with other countries, countries in Africa as well, and also collaboratively with Bangladesh and Thailand and Malaysia as well. So we look forward for that, and definitely we will try to identify the opportunities, how we can actually collaborate on this. Because for me, I have also visited quite a lot in Thailand, in the coastal areas. I have seen their hatcheries, the hatcheries which is quite advanced, that is from the DOF or Department of Fisheries side. There are also the conventional hatcheries that you mentioned. Initially, I also mentioned that there is a huge demand of the swimming crab in this country. And actually for swimming crab, they also have a hatchery, which is not they call hatchery, it is called crab bank, which is interesting topic they are using. But interestingly, the methodology is almost the similar. They bring the gravid crabs and they allow them the natural environment, or they mimic or they create the natural environment for the swimming crab, and then they let the swimming crab to allow. And then, of course, when the swimming crab is hatching, then they release this hatching to the sea or back to the sea. But of course, we understand the survivability is always an issue. And I think it is likewise the same for the mud crab that we can understand from your presentation. So I see a couple of other comments from my colleague like Dr. Bahul, from participants like Noha, Elbana, Ahmed Jalal, Othman Bozo. They have basically sent their special gratitude and compliments for your presentation. A special thanks for your presentation. So with that, I believe it was a wonderful and very long webinar, and I am particularly very pleased and happy that many of our participants are with us for more than 2 hours, which is something remarkable. And that is only because we probably have made this deliberation interesting for them. So we like to convey our special gratitude and thanks to all the participants who are listening and who are still with us and who are with us for last 2 hours. Please feel free to communicate with Professor Badrul Munir or with us at AIT for any kind of need that you may think that can help you to develop these sectors, to develop your research, to further go ahead with your project. So with that, allow me to say thank you once again. Thank you very, very much. And have a good evening, good afternoon, and good morning to all of you. Dr. Badrul, would you like to say anything?
Thank you to Dr. Zakir. Actually, Dr. Zakir is my university friend. university friend, and he also do in AIT, and I also just after 1 year I also completed my masters in AIT. It's a very much good opportunity for me actually. If you interested to do this, because conventional hatchery takes lots of money include. So if you have a pond, if you maintain the salinity, you can do this system. You can do this and harvest the mud crab and then sell it to others. This is farmers level. It's a very good and profitable project, that one. So if you need further my help, I always ready. And I think AIT extension, I also know about AIT extension is is for the pupil of the pupil, AIT extension. So I would like to request AIT extension to collect all the interested person and then communicate with me and my team, especially field level experience more about Dr. Abdul Hanan. He 24 hours, 7 days in the field. And so that we are a team. You can contact with us. You can contact the IT extension directly to me. We can help. Inshallah. So no worries, and thank you. And just say my salam to everybody, and then good day to everybody. Okay, thank you.
Thank you. Bye bye, everyone. Bye bye.
Bye-bye.
Bye bye. Bye. Thank you very much, Dr. Munir. We will be in
Thank
touch.
you.
We'll be in
Thank you.
touch.